So good morning, everybody. Myself, head of the Department of Genetics and Plant Breeding. I welcome all the staff members, students who are online and offline for this lecture to be delivered by Dr. Sudhir Paul Ahlawat from NBPGR. He is a principal scientist and ex-head of the Division of Plant Exploration and Germplasm Collection at the National Bureau of Plant Genetic Resources in New Delhi. And he has a lot of contributions with coordinating and helping in the collection of germplasm, and he is also presently implementing 3 externally funded projects, including UNEP, then again along with Uzbekistan, and also he is a principal investigator of several projects. He has conducted more than 20 explorations and collected 1,000 accessions throughout the country. And he is also the task force joint coordinator for the National Mission for Sustaining the Himalayan Ecosystems. And he is also the nodal scientist for agro biodiversity for ICR at NRM division. And he is also the nodal scientist for 4 institutes, CAFRI, Kazari, Jodhpur, and ICR, NEH, Northeastern Hill region, and also Goa. And he has published several research papers, and he has got several awards, Brandeis Prize, Best Worker Award of Caffrey, then again, member of the Regional Variety Testing Committee. And he is also taking classes for the postgraduate students of PGR, and in the course Advances in Explorations and Germplasm Collections and in situ conservation of PGR. And he has also guided more than 6 students, and he's member of several societies of agriculture related, and he has also contributed several trainings and workshops. So with this brief introduction, I welcome Dr. Sudhir Paul Ahlawat for delivering the lecture. I am very happy. We will all be benefited by the lecture because of he has got a vast experience in his area of plant exploration and collection. So thank you very much, sir. We welcome and we will start the program. Thank you, sir.
Okay, sir. Thank you very much, sir, for giving this. But these assignments were when I was in ICR headquarters, task force, etc., and the coordinators. So 2 weeks before, I was having the division plant exploration and plant collection slide. Okay, and this was a lecture we have presented at FAO also, so crop diversity for sustainable development and bridging the gaps between in situ and ex situ conservations. So the outline of the presentations is the crop diversity for sustainable development goals, and crop diversity for sustainable agriculture, challenges for food and nutritional security, crop diversity ex situ status, international and national, approaches to find the gaps and to bridge the gaps, some initiatives and achievements, and policy interventions required to bridge the gaps. So crop diversity for sustainable development, that's very important because the nature has provided enough diversity to fulfill the requirements of human beings for food and food. But as Gandhiji has told, it's not enough to meet the greeds of the human beings. And you all know the anthropogenic developmental activities, particularly the agriculture land area is shrinking. For us, our density and area is also shrinking. So we are losing a lot of diversity, and particularly for the adaptive and evolutionary process, that is also the international level very important, being focused nowadays. So synergistic coexistence is very essential for the industrial scientific exploitation. So coming to the sustainable agriculture, that's the integrated system of plant animal production system that should be the long run enhance environmental quality and based on natural resource base. That's the plant genetic resources are natural resource base upon which the agriculture economy depends and satisfy the following 4 essential goals: human needs, enhance environmental quality, sustain economic viability, and quality of life farmers, farm workers. That's very important. So there is a relationship between people and nature which is very complex and very not to be managed at this social and ecological systems in isolation. We have to synergistic approach. We have to maintain that, and both should evolve and allow the changes also. See, you all know there are so many crops, so many species have evolved in the nature. That's a number maybe in the millions. But if we look at our scientific breeding program, scientific activity, there is very limited. And even when we look at the number of these species developed by artificially by the human beings, that's maybe 2 or 3 only. But the nature has evolved so many by variety, diversity. So organic changes, adapt, innovate. That is the basis of the future breeding program. Then history also important, and that place matters also. So for resilience, social and ecological resilience, the human activities are very strong drivers of the ecosystem change. People can do act as stewards of their environment, and well-being is supported by the ecosystem only. Coming to the Sustainable Development Goals, there are 17 goals with 169 targets. Among them, these 6 goals, SDG goals, are very important and related to the agriculture point of view. Means that the mainstreaming of biodiversity for increasing farm productivity, reduction in input costs. Means no poverty, then zero hunger. Then they should ensure sustainable production and be responsible consumption and climate resilient PGR reduction in methane emission. So these are the climate related actions. Then third one is the good health and well-being. That's through the improve nutrient content, quality, and reduce post-harvest losses. That's very important one. For life on land is important for on-farm agroforestry conservation. These are the SDG goals which we have to take care of. So that these our activities in the agriculture we are taking care of. There were Aichi 2020 targets. That's also stress upon understand the value of the biodiversity, mainstreaming of the biodiversity, then sustainable productions, then reduce invasive species, then conserve gene pool, enhance resilience to the environmental challenges which are taking place, climate change implement Nagoya Protocol, and respect and conserve the ITK. So these are the activities, these are the goals, targets which we have to meet, and so the genetic resources. So in this regard, there was a first International Agroforestry Congress were held, which is come out with the Delhi Declaration, which emphasized on complementary conservation strategies, awareness and capacity building, and exchange instruments, and biosecurity, frontier technology for sustainable use, traditional knowledge, participation of private organizations and society, ecosystem services. Agroforestry Index was also talked in that. And the Biodiversity International is working on that, including for India. And paradox of that, then there are challenges that there were a large number of plants, nearly 3 lakhs plant species, and out of that, 30,000 are edible and 7,000 are used as food at local level. But we have narrowed down them to the 30 plants, crop plants, which provide 90% of the calories. And only 3 crop plants, rice, wheat, and maize, provide 60% of the calories. So that is the major challenge of the nutritional security and malnutrition. And that is causing the adversely affecting the human health also. And our food basket has reduced instead of diversified basket, which we used to have earlier for long back, but now we is limited to these rice, wheat, maize, and potato 4 crops. Another challenge is the growing populations. There is no policy to control the Indian populations. I will say now. Earlier it was long back in 70s, but hum do hamare do was there, but I don't think whether in practical level it's there. So by 2050, our population will be 1.66 billion, and then it will start declining. So we have a challenge to feed the 1.66 billion populations. For that, we need to increase the production nearly by 70% from 2050 to 2015 base years. And the plant genetic resources, for which I will be talking, and we are here, that will be the key to achieve that. Another challenge is that in the climate change. So global reports indicates 10 to 40% in crop production by the 21st century because of the impact of the climate change. And this color is representing to the India. There will be more than 40% change and maybe loss also. So that's the challenge we are facing and we have to face. Water availability will also reduce by 30%. The risk that is also the risk that will also reduce. Another challenge is increasing homogeneity in global food consumption. I highlighted in other slides also, slides was 1960. We have shifted to the relative importance of the crop in our diets, and hence are more dependent on fewer crops, widespread, less instead of the widespread crops. So these wheat, rice, and maize have come to the food chain system increase more. But if we look at the compare other crops, that these are the having higher ratios in terms of supply to calories of the food supply. But these are supplying the less calories, and their contribution in the food is more. Consumption is higher than other these crops also. So now we are focusing on the millets. That's why this year we are celebrating with the United Nations Millets Year. Scale of challenge, if we look at the 2 billion people lack the key micronutrients like iron and vitamin deficiency, and 155 million children are stunted like that, some are devastated, and adults having overweight, obese, etc. And 88% of the countries face serious burden on either 2 or 3 forms of malnutrition. And for that, the only solution is the nutritional security, for which the millets are being focused now. So thus sustainable agriculture and diverse food intake are equally important for healthy population and for the crop diversity for both. Because if there is no consumption, no demand, then the diversity, there will not be the crop diversity. If there is no demand for the millets, then why the farmer will be cultivating? So for healthy population, for health, the crop diversity is equally important. There should be demand for that. That's the key basis for that. So to meet these challenges, food and nutritional security in the changing climate, germplasm is the key. And for that, we have to go for the exploitation through novel pre-breeding strategies using crop-wide relatives and the fundamental reinforcement of the entire plant breeding chain, vital for sustainable system of global food security. And when we talk to the pre-breeding, intra-specific hybridization, inter-specific hybridization, intergeneric hybridization, these are, we have to use these techniques. We have to explore them, because in many times we don't get the suitable genes in the same genus. We have to go through intergenic process. But nowadays the gene biotechnological techniques are available for the gene transfers, and that has been GM crops are being advocated and being replicated in the country. And that could be another method by to overcome, which can overcome these conventional breeding methods. So PGR is well known, but to the students, I would like to tell that this includes the tissues, pollen, DNA molecule also, and containing functional unit of heredity. That is most important. Means there should be a germ cell which should be reproduce the plant to have some genes, full complete genes which can be used. Otherwise, that's of the no use of the DNA molecule. It should be a complete gene. So coming to the diversity, as I outlined in my presentation about the India, it's a gene-rich country, having a lot of diversity in the plants, animals, etc. over there. That will coming to the plants. 817 species of wild crop plants are occurring there. So coming to this genetic resource management system in the country, there are 5 genetic resource institutes. Among them, that includes the animal genetic resource, agriculture important microbes, also fish genetic resource, animal genetic resource, and the plant NBPGR. I'm talking about the plant genetic resources management system. The plant genetic resource systems includes the collection, that is the primary activity. From there it starts characterization, then it goes to the conservation, and from conservation and after conservation it is used the utilization. It's very important. So collection is for utilization finally. So coming to the diversity, it was an Babilo who proposed the 8 centers of this diversity in the world. He traveled across the world, but unfortunately he could not come to the India. But despite of that, he gave this second Hindustan Center of the Diversity. He also recognized India as one of the major centers of in the world of the crop plants diversity. And after this Javan Jokoski 78, they also have identified 12 among the 12 also. This Hindustani region is also a very important center of the diversity. So that consists nearly 170 crop species having native diversity in the regions. And these are given the primary centers of the diversity that includes rice, etc., pigeon, pure, bean, cotton crops, like that. Then the regional centers of diversity, wheat, barley, etc. Then secondary centers of diversity, which are of African origin, like sorghum, palmate, fingerlet, etc. Then tropical American crops, these maize, etc. So it's not only that the crop originated in India are having the enough diversity, the crops have come later on to the India also have sufficient quantity of the diversity like maize, etc. in these crops also. And the presentation where thesis by Washi took that was introduced this sunflower in 1960. So what is still there are good. So coming to the PGR crops, since I was heading this exploration collection division here in NBPGR, so we have to deal all the crop plants. So we have divided it that entire crop diversity 581 into the crop groups. 12 crop groups we have divided, and that the diversity in India that we have. So here we have given the cereals 4 major crops, so 6 crops, 16 crops like this, 17 crops like that we have divided here, and the major crops are also given here. So this is the diversity we are dealing with at the exploration division or NBPGR with respect to the cultivated crops. So the task and work is huge, enormous. But among them, 183 are indigenous crop species of economic importance. That also we have to cover for collection, exploration, conservation of the germplasm. Coming to the importance of the germplasm, it is well known that there are so many germplasms basic as collected, but they have been released as the varieties. And here are the given, but particularly in the perennial trees, like the fruit trees in mango. If you look at here, so mango, citrus, banana, there are so many varieties directly collected germplasm has released as a variety. So this shows the importance of the germplasm. And in other potential crop and so many other crops also there. Maybe if you look at that, there will be several. And when we study the plant breeding, that the basic selection, the selection is the first method of the breeding method. So that has been used in such cases, and simple selection from the populations, these varieties have been developed and released. And NBPGR is also registering the genetic stores of trait specific accessions, which stands now 1049 and 183 crops have been identified. These are the character trait specific accessions have been registered. So there might be several from the TNU also. So coming to the importance of the genes, then there are several genes which are saving crop from epidemics and abiotic stresses. So growth stunting virus have threatened the entire rice crops. So for the gene transfer Nivara, Odisha, that saved the rice crop against this virus. So downy mildew resistance saved the muskmelon crop in US. That the sorghum also resistant green bug insect also. So there are several genes, and for this submergence tolerance genes found also has been from the material collected from Odisha by the Agri, that also found. There are so many genes in the Indian origin are being used world over. So that they all go, we have found out, or the other countries are finding out from the germplasm what we collect and conserve. So that's very important activity. Coming to the exploration collection missions at NBPGR, what we have been carrying out, that there are four type of missions we execute. The first is multi-crop explorations, that region-specific explorations, mostly in those areas which are less explored or underexplored, and having the diversity-rich areas. Mostly nowadays, these are the rain-fed or tribal areas, remote areas or disturbed areas where these such kind of explorations we are carrying out. Then crop and trait specific missions that we undertake in the larger areas and based on the demand of the plant breeders. Then rescue missions we undertake in the where there is the areas are prone to cyclone or something like the catchment areas of Sardar Sarovar dams are being conducted. The areas like Tehri Dam area like this, these are the rescue missions, which are the areas going to be submerged or something like that. Then earlier, before the CBD, international missions were also carried out involving other countries, but nowadays, after coming to CBD 92, these are not possible because each country each had sovereign rights on their genetic resources. But I'm having now a project in from the Uzbekistan DST supportive project with the Uzbekistan as PI, in which the germplasm is being exchanged. We are also giving them and they are giving to us. So it's an exchange project with the Uzbekistan. I'm looking for other countries also to have the germplasm of our use. So this is a total, in nutshell, this is the status of the germplasm, current status in 2023, at end of December 22. Total 2,800 explorations we have carried out by this division exploration, to which I am representing here, and nearly 250,000 accessions we have collected. That includes cultivated ones and wild ones also. Earlier the share of the wild relatives was less, around 4%, but now it has increased to 26% in recent explorations. So this is graph is showing the total ratio of the germplasm what we have collected, and that cereals are maximum, followed by pulses and vegetables or seeds also. And this is starting from 1976 when NBPGR was established. So every year what are the number of explorations cultivated. So we have this graph is showing that this is the period when this NHP project was going on, which has very significant contribution, which has been nearly 1 lakh accessions to the gene bank. So that's very important period. So such kind of mega projects are important, very important in now also, because maximum now we conduct 30 plus minus 2 explorations and nearly to 2,000 to 2,500 accessions in the gene bank. That's our limit. So such projects, even single project have added to 1 lakh accessions. So that's very important such project. So this is the current holdings of the gene bank in different crop groups. I will skip this. This is can be seen on the website. But this is the second largest gene bank in the world, having 0.46 million accessions in our gene bank. Maximum among the maximum are the cereals ones, then the legumes, then vegetables like that. These are there. The material which could not be conserved in the seed bank, that are conserved in this tissue culture form or the cryo bank, that's also beneficial. So field gene bank conservation is very important. These are the material which cannot be conserved in either of the form. That's a tissue culture, but tissue culture is again prone to the disease and pest. So is the field gene bank also. But another thing I would like to share the learned audience here, that please share the accessions, whatever the material you have in your field gene bank, so that it will come into the national database, take the IC numbers, and the duplicate collections, etc., can be saved, or the material, if demanded even in the TNU by or some other state, can be shared with them also, so that can be used widely. So, NBPGR freely provides to the material work. So coming to the planning, exploration, and collections, we get the demands from almost all the state agricultural universities, as well as crop-based ICR institute, for which we prepare every year exploration plan. And for that, we send the demand also to them, and we receive, but many cases we receive the demand in terms of the state, many people said there are several states which are not possible, or several number of districts in 1 or 2 years to be covered. But if you look at, only 2 to 3 districts can be covered in 15 days explorations. So, and no scientist wish to go for live away more than 50 days from his family or away from that. So when this purpose of showing this slide is, if you are planning, want to join us or want to take up any exploration, so that it should be specific in the particular targeted area, 2-3 districts only, maybe first year 3 districts, maybe second, third, fourth year, like that, and should be crop specific also. And the period also very important these days. So when sending the proposal, send it in this format properly. Then, as I have shown, more than 500 crops and 50,000 species, including medicinal aromatic plants. So we have to prioritize the crops and the species based on the economic importance, immediate need in breeding for trait specific, threat level, RET specific, rare threat species area, or area which is vulnerable, and conservation requirement. There are several species in NBPGR also which are not even representing in the gene bank also, or not worked also. So we have taken up that we are finding, and we look for your suggestions also from your learned group, that please send us the information, or that these are the species crops which we should target, we should prioritize in our exploration program, which are the areas we should focus on that. So coming to the approaches of bridging gap between in situ and ex situ conservation, the first one is the georeferencing of the whatever the data we have. See, prior to this getting of this GPS, geographic positioning system, we were not having recording the latitude, longitude. Now we are what we have done, which the exercises which we have been latitude, longitude, that have been georeferenced. So around 62% accesses have been made, 1.93 lakh accesses belonging to 1500 crop species, and this is a link for that at the NBPGR database, which you can see. And by there are several apps which will come in the next slides. Yeah, so these are the PGR portal, then PGR claim, the genetic resources, Vigna and other species, then herbariums, etc. So this is a website on which you can see there are just several genetic resources, this gene bank dashboard, PGR claim, genetic genomics, so profile data also. These are the things are available on NBPGR website, as well as can be used on the mobile apps, iOS or Android based system, by which you can know the actual status of conservation collection, as well as from which area this material belongs to. So another activity in my division was to identify the gaps. So we were taking each and every one crop like that. So far, it lasts maybe 7, 8, maybe 10 years. 21 agri-horticulture crops have been taken up. That include rice. These are the crops we have given. And we have taken the data from the database, and then we have analyzed. So if we look at the rice, nearly 1 lakhs, now more than that, accessions have been collected. Locations you could find 5,000 having the location. 55,000 were in sites for which the sites were unknown, and out of these, only 37,000 were in the gene. Of course, the number of gene means the 37,000 among these 44,000 were having location details. So we have found out the gaps for recollection. That's nearly 7,000 accessions. So this is activity is being carried out in my division, in our division. So where this much crops we have completed here. So this year by year we are taking up this activity. So then these are being plotted on the Indian map of the country, and each dot represent each accession. If we point over here in that database of the NBPGR, which I have shown over, so it will show the area, the name of the place, and the IC number also from which it has been collected. Like this, the maps are being uploaded on the website. This is the link also for that. Coming to the Tamil Nadu. So I have taken the data from the database for the conservation research. So the database revealed nearly 14,000 accessions have been collected from the Tamil Nadu state. So that includes 800 crops and species. But among them, the conserved are nearly 6,000, representing 289 crops species. So if you look at the gap, that's more than 50% in both the cases. And top 21 crops which are collected and conserved includes the rice, sorghum, Sataria, Jatropha, Illucina, Caginus, Lab, Solanum, like this. And when I look at the Vigna Mungo, 192 accessions were collected and only 4 were conserved, so 188 is the gap. Like this Vigna radiata 89, 60, 73, these are the gaps. So this is a very grim situation with respect to color conservation. So I feel that Tamil Nadu still there is a lot of chance to conduct explorations, collect them, and that is possibly only involving the TNAU. Because we have the stations over here, the Thrissur as well as Hyderabad, and they have to also focus them. Though I have been trying in my service has had it last 8 years, but few explorations have been undertaken in Tamil Nadu, but still more is required. Then coming to these 21 from the 21 crops which were analyzed at my division for this analysis. So among the 19 crops I'm presenting over here. So these are the much accurate figure, total accessions collected, the latest one figures. Then accessions collected from Tamil Nadu are 1,005, the conserved 700, 341 is gaps. And there are 38 districts in the Tamil Nadu. Out of the 38, 30 material was collected from 32 districts and conserved only from 22 districts. So these two show the collection from 11 districts and conserved only from 3 districts. Like this, it is the shown over here. So this is a gap base for Tamil Nadu. For this presentation, I have made this analysis and being presenting in front of you today. For this lecture, I especially made this. So coming to the another activity, approaches to find the gaps also. We are georeferencing diversity mapping for this species richness and diversity map of 16 wild species of Solanum in India. This is wild species of Solanum. These are the 16 species listed over here, wherever they are from collected, and this is also shown here in India. So total we had nearly 1,000 accessions, and less than 20 accessions which state are being given are not given over here. So this is a different species, wild species of the Solanum are presented in these presentations. That's the way we are doing it, analysis, gap analysis, for planning further explorations, collections, activities. Then coming to the crop viabilities, that is very important component of the plant genetic resources. See, you all know very well, when agriculture started 10,000 years before, it was all wild plants, and hunter-gatherers, our first farmers, they started gathering seeds, so growing, sowing, domesticated and improved, and now we have a good number of cultivars, varieties, crops which are edible and like that. So this is definition. Plant taxa have indirect use and derive from its close genetic relationship to a crop. So they are very important, needed much more than ever before to meet challenges like food insecurity, climate, and survival in general, but their survival in the threat for massive and unprecedented destruction of nature around the world. So coming to its uses, collections, and prioritizations. In the Crop Science Journal, this was published in 17. There are nearly 4,000 uses are documented of the crop plants spanning 127 crops. Among them, 3 crops were major: sunflower, wheat, and potato, which are these maximums have been used. And top regions for using was CWR and breeding flower, biotic stress means pest resistance and drought resistance and seed size yield resistance. So there is a need for novel genes to break the barriers through the introgression and through the bioavailable tools also increase pressure wild species that's increasing. We are losing them. So there is a need to collect them because there is a very meager collection in gene bank which comes in the next slides, which I will show. There are only around 4% collections in the gene bank belong to CWR of the total collections of that, and among them maximum are of Nivara, Rufipo, Malabaricum, Cucurbits, etc. Momordica, etc. These are the species. So we have prioritized them. And the criteria were based on the economic importance of crops per se, level of closeness to crops, possessing traits of breeders' interest, extent of distribution or threat to them, and seed storage behavior. Based on that, we have prioritized the collection of CWR crops. So this is 13,479 accessions of 1,100 wild species of PGR of importance are conserved in the gene bank of the NBPGR. So among them, 238 species we have prioritized based on the criteria I explained in the previous slide for the belonging to the 80 crops have been prioritized. Among these 200, we have further narrowed down to 100 high priority, 116 sections that includes the wild, semi-wild forms, progenitor of these. So this is the crop groups, and for 13 crops, we have identified 73 genera and 83 species like pseudo cereals, 3 major crops, for which related wild traits we have identified 40 species, then grain legumes 58 species like this are given. So for 168 crops, we have identified 141 genera of 833 species. That's the work. And this is the work we have recently published along with K. Pradeep, belongs to your state. Presently, we are working at Tirchur, and the book has been recently published as well. It is available on the NBPGR website. Download CID, download. This is also this you can download, and more lot of details you will find out the crop wild natives in India from the NBPGR website in this book, which very recently 21 we published this book. We have identified the areas also for the exploration plans that starts. These are the areas shown over here: entire Himalayan range, including northeastern range, the desert area like this, Vindhya and Satpura, western Ghats like that, in the eastern coast along the Tamil Nadu, Odisha, Andhra Pradesh. So these are the areas we have identified. So these are the 11 areas we have identified for CWR providing exploration collection. And this is the approach for bridging gap between in situ and ex situ conservation. First, we have to diversity, then identify, recognize the custodian farmers, improve the access availability to the diversity, and assure the benefits to the farming community as well as the good practices to the community. The benefits, sharing of the benefits is very important. If we take the material, use their material, then it should be important. Benefit must be shared to them. There are several strategies in piloting good practices that reduce risk, increase productivity. Particularly, we have to develop the material or use the material which is abiotic tolerance, which has abiotic tolerance. But our farm, where this farmer varieties, land days are very important for that. Very good material for that. Person. And I have to increase their access and information of material to the farmers, identify best material which is suitable for the market, preferred in the market only. That material, otherwise if there is no demand in the market, then there will not be no use of cultivating, growing that, and farmers will be in loss. And we have to increase the demand of the material, that policy, these will come in the subsequent slide. So I'm implementing the UNJEF project from the last 6 years. This is going to end on this 31st March now. The project is mainstreaming agrobiology conservation utilization agriculture sector to ensure ecosystem services and reduce vulnerability. This is a mega project, and these are the 3 objectives: adaptive tourism, crop diversity, resilient agriculture, livelihood strategies, policies, improve agriculture support system to the institutions, framework, partnership, etc. This is implemented in 4 agricultural regions using 20 crops. These are the area districts, nearly 17 districts, starting from this Himalayan region, the arid zone of the 3 districts, then central India, as well as of these are the crops or given here. I will, in brief nutshell, our approach to mainstream the agroforestry was enhancing crop varietable diversity, repatriating aceto collections to the farm. Repatriating means taking from the gene bank and to the farmers. Land race we had from these areas, we are repatriated to the farmers also, gene banks. And in this project, we both NBPGR, gene bank, as well as farmers, both way. It was a reciprocal approach, giving and taking back to the gene bank. Both have benefited in this. Enhancing accessibility to diversity seed, developing community seed banks, very important activity for that to enhance the diversity, sustainably maintain the diversity, ensure to the farmers. Then enhancing livelihood options, income through add value options, entrepreneurship development market. This was another important milestone activity on which I had doubt when we started this project. See, we are talking to the cultivation of the to the learning group. I would like to say when we were taking this project, we were talking to cultivate the landrace farmer varieties, which are having half of the yield as compared to the hybrid improved varieties. So farmer will be the loss of the 50% loss, half of the loss of yield of the value. And MSP and dates are the same, whether you take the hybrid improved hybrid variety or the farmer's variety land dates to the market. So why the farmer to cultivate, grow that? For that was a problem, and I thought what we have though we taken that it would be very difficult to implement this activity. But in subsequent slides, I will show how we have been able to successfully market this. Then capacity building, awareness, dialogues, etc. Consumption, that was also important activity. Our approach was the crowdsourcing participatory variety selection with the material which we have taken from the gene bank, etc. that we tested on the farmers' fields, and it have enriched the variety recommendations, improve on farm testing, and engage the empower the farmers. So crowdsourcing was mainly if it comes. So what we done, we have taken the material from the bank as well as this belonging to the same area. We have multiplied it. We have given it to the farmers, giving them randomly blanket without giving the name of the variety. And each farmer was given 3 varieties to grow along with its crop, using its own practices, same to the farmers. And then so in the first year we took 20 varieties, then farmers have selected then among them 1 or 2 varieties. Next year again, like 3 years we have conducted this trial. So at the end of the this testing trial, we custom these champion farmers and all these farmers using that one. We have narrowed down to the 2 or 3 varieties finally. So coming to this, nearly 3,000 land disease farmer variety 24 crops were collected and repatriated from the gene bank, evaluated through participative variety selections, and then for which the nearly 500 mother trials for those trials in which all the varieties were grown by the on the either of the farmers field in the field of champion farmers, these champion farmers, or more than 3,000 were the baby trials, means 3 varieties per farmer each year were given to test their varieties. 161 varieties were identified as most potential for large scale seed multiplication. So among them, nearly 400 farmers have been trained, designated champion farmers. So 139 self-help groups were established, and 1,700 farmers have took membership of that one. And we linked them to the FPOs, farmer producer organizations, or the groups are 25 to private companies for value addition, product development, and tourism. So several workshops were organized, but important thing is the linking. Not only this FPOs cannot do nutritional profiling was very important. We have to tell to the users, consumers, why we are providing them. We see what I told that in the beginning that the 50% loss, how to beat that. That's so that this nutritional value of the varieties, which we have to tell them, and branding, labeling, etc. All this. So we have to sell the material double the rate. which is prevailing in the market. We have to select such varieties which are having nutritional qualities better, so that only will be so sellable at the higher rate. That's what we have been taking out. So we are developing the community seed banks also, like this also is 29 community seed banks have been initiated, and they are maintaining more than 2,500 traditional varieties of different crops. Some are mentioned, or the number is quite high. So like this. So another approach to spread the local disease farmers also was this char dhania approach. From Chhattisgarh it is there. They spreading best variety using char dhania. Each farmer spreads the seed to the 4 farmers, sharing at mass level like this. That one of the another target of this project was to maybe more than 10,000 or lakhs of the farmers. These materials will go like that. So this is another. And coming to the custodian farmers, very important activity, which maintain the rich diversity, self-driven by conservation, having wider knowledge, also highly motivated, self-recognition from the community there. But the issue is how can we consider their role as conservator, dynamic inventor, promoter of diversity? Can such customers be recognized as community gene bank and publicly supported? Or what are the policy? What for they have to do? Whether their generations would continue to that? So these are the several questions coming to that. This Prahlad Sahu from Chhattisgarh, Dhamkari district, I visited his farm also. Very poor fellow, and having very kacha home also, was maintaining 268 native rice lands and maintaining the farm, this plot similar to the scientific. We research institute at the universities maintain them, but no input, no benefit, no support from them. Similar was the Deepen Parva in Assam, conserving 89 native rice land disease. So compensation to custodian farmers is very important. Why? How long they will continue with their passion and all these things? So Dr. Paul raises all of the times this question, this issue in his speech. So linking agroforestry with farmers' livelihood, that was very important. So these are the value chain development for heirloom rice Assam. These such kind of the initiatives, entrepreneurship, value addition, enterprise branding, labeling, etc. have been developed, and now we are successful. We could do this at a mass level. So 12:30, I have to finish now. So another was Jeera Fool from Chhattisgarh. This has also got the GI tag, and its production has increased a lot. Now nearly 1,000 hectares. It is few years back slide. So total production has also gone quite high. So these are the benefits the farmers are getting nowadays for implementing this such kind of project. Then underutilized crops, that's a mitigate impact of climate. These are very important, but their area and diversity is increasing at alarming rate, particularly in forests also, and the trees are most vulnerable. So these we have to also look at and there is no, when I look at the research institute of the horticulture, they are mainly concentrated on the major, most important crops, fruit crops only. But there are very few institutes, or the scientists are interested to work on these such kind of crops also. We have to look at them also. Many times we don't have their conservation protocols also, particularly those are very highly recalcitrant species, very high moisture content, cannot be desiccated. So in NBPGR do not have the conservation protocol. Say for the Garcinia also, we do not have the protocol for that. For the CGGM jamun also, we don't have protocol. Mango, so many crops we don't have protocol to conserve them in the crop bank. So these are the things which we have to target. Then there are several international conventions like CBD, Cartagena Protocol, then PGRFA, ITPGRFA, Nagoya Protocol. We are bound to conserve this diversity, for which we have geographical indications of the registration for protection of that act, then BDA, Biodiversity Act, the PFRA. They are effective from that. They are conserving. Then there are several agencies which are utilizing the PGRFA, conserving utilizing speech plant resource. For sorry, this is PGRFA. Sorry, this is a mistake. And implementing the sustainable goal of the United Nations FAO, that's the farmers, the institute, ministries, protected areas, then CWR, wild edible plants, MNDP, and then NMPP, so many. Several R&D institutions like NBPGR, then crop-based institutes in MTS, FGB, SAUs also, BSI, CISA. These are the agencies conferring them. So coming to the farmer PPFRA also. How to incentivize these custodian farmers, those or the for communities which are conserving in situ conserving these varieties. So they have come up with the awards, rewards, or recognitions also. So that there's a plant genome savior awards, community awards. This is 10 lakh rupees. 30 have been awarded like that. So this is another thing. But then farmer reward maximum 10 consistent settlement cash rupees of 1 lakh. 50,000, maybe 3 lakh now, and 1 lakh also. So the important questions arise after that, that once they got the award, that's a one-time award, they got it and they have been recognized. But what next? Whether is it sufficient to sustain them, continue to conserve them? And what's the use of that when conserving after so many years to the farmers means benefit saving I'm talking of that. So these are the community awardees. If you look at here, the present our agriculture, honorable agriculture minister Tomar ji and previous minister Sri Radha Mohan Singh ji, honorable our MS Swaminathan ji here, or here the farmers are here. So they are being, they also feel very, everyone feels happy when they get the award. But what next after that? So we are also conducting at NBPGR using the schemes, tribal schemes, SCSPs and schemes. These are conservation awareness programs. I was handling these two schemes previously. Several programs we have carried out, but we also go and tell them, but there should be some concentrated policy program to do such activities. So coming to the seed networks, I was talking about the community seed bank, why it is important. This Indian seed network is only for improved varieties. They do not target the farmer varieties and the land races also. So there are the institutes, CAU, SAU also there, NSCs also there, seed corporations, seed agencies also. There are so many institutions are involved, but only for the improved varieties. Then farmer varieties are not in the formal seed supply systems. I think now the DAC Ministry, Agriculture Farms, are now considering them, and in the revised seed bill, they are considering, I think, to consider them. Then access benefit setting to farmer for their variety is yet to be realized under PPFR Act. This is also very important question. I don't think any incentives or the benefit accrued from the conservation for the registration protection of these varieties that have gone to the farmer. There are not even a single rupee have reached to the farmer. Then policy support to the custodian farmers conserving large number of varieties for their passion is very, very important. Then establishment of ex-situ in-situ gene bank for conservation of crops, CR diversity is very important. Ex-situ in-situ gene parks for RET species, overexploited species, medicinal, that's also very important issue which we have to take up. And higher minimum support price for the farmer varieties and traditional land races due to their better nutritional value. That should be ensured. Then only the farmers will be continuing. See, world over they are talking of the in-situ conservation to continue the adaptive and evolutionary process. Because the material kept in the gene bank remains static genetically. And everybody wants that as in the nature has evolved so many number of the species, human beings, animals, crops and plants that should continue. For that, the conservation should be in situ. But how? Who will go conserving them without any policy support? That is the major question. So that was the previous slide. In the previous slide context, I would like to tell you that I have taken a project recently this year, financed also from National Rainfed Area Authority for the conservation of the wild rice or Pogon in Assam, and it was going on very well from past 3 years. When we selected, we identified that it was a pure crop pond, more than 1 acre, and going on very well. But the involvement of the community is very, cooperation from the community is very important for that. And what they did, this last year, they grown some fish in that, and that has significantly reduced the population of the wild rice in that. So community, there are so many challenges we face in such kind of conservation, community-based conservation program. So thank you very much. This was from my side. So I listen, and how can I unshare? I don't know how to unshare this.
Now the topic is open for a discussion, I think.
Yes.
I'm happy to announce that our Dean School of Postgraduate Studies, who is mainly responsible for organizing such these lectures, is with us. So he will start.
So thank you, sir. Thank you for sparing your time and enlighten our people on the NPPGR activities and also your wide coverage on the different conservation activity, not only the crops state-wise and the crop-wise and district-wise data you given. It is give us more view on how the Tamil Nadu also contributed on the gene bank. And another thing, this presentation definitely enlighten our students on how the national system which help to conserve the germplasm, and also new initiatives which you given on different for example traditional varieties they conserved, and also the farmers recognition, everything you given. So this will definitely our this CPPG also will motivate the farmers to apply some of the awards and other things. So not only the evaluation for having the students, but also as far as our request, you agreed to give the lecture, guest lecture also. This in this Tamil Nadu Agriculture University highly thank you for accepting our invitation and give your lecture on special lecture for this occasion. Thank you, sir. So this is my small remarks on that. Now I ask the students to at least ask the few clarification questions maybe we expect from the students, and also the online participants, please raise your questions or raise your questions in the chat box that the speaker will answer. Thank you, sir.
Okay. Sir, I could not. Can you repeat, sir?
I will repeat. Sir, we are now making the collections along with locations.
Collecting the accessions along with locations.
Locations, yeah.
Yeah.
Yeah. So after certain years,
Okay.
in the some changes may take place.
Yeah.
Yeah. How we can revisit the area, any program with NBPGR to revisit after a certain period in certain location?
Yeah, that is possible, but we have to see the how many years before that material was collected from that year, and if the same we are going for the same material, means the same variety or same land races. Well, if there any changes have taken place, there should be sufficient time, say 15 or 20 years like that minimum. So that some changes in the population would have taken place, and that we collect them, then compare them. That would all see adaptive and evolutionary process goes on. And even in self-pollinated crops also, we know 3-4% breeding also takes place. The farmers use mixtures, etc., for the safety, for the environmental safety point of view. So definitely that population varies. So the 50-20 years, definitely we should revisit. It is already known to us, and we always say in our discussions also, as well as presentations, that revisit after certain period is essential to collect that material again.
Propose formal vote of thanks. First of all, I thank our Honorable Vice Chancellor who permitted us to conduct this guest lecture series. And I like to thank our Director CBPG and Dean SPGS for their constant support and guidance in conducting this event successfully. And it is my pleasure to thank our speaker, Dr. Sudhirpal Ahlawat, Principal Scientist NPPGR, for his thought-provoking, informative, and interesting lecture. Thank you very much, sir. And I thank all our head of the department, genetics and plant breeding, chairman and advisory committee members, and the professors and all the student friends for their support. Thank you very much. Onada.
Okay, thank you very much to all who have joined and listened and given me this opportunity to present this work before you. MS Swaminathan Foundation is doing excellent work in this aspect. So we also, research institutions also should take up this activity. That's what my purpose to only to enrich the knowledge of the students as well as researchers also. Thank you. So can I leave? Okay,
Our
buddy.
student has to ask one question. Sir.
Yeah, please.
Morning, sir.
Morning.
Sir, I should tell that we are facing so many climate changes increasingly nowadays. As you
Yes.
tell,
Yes, continue.
we have to mitigate that climate change. As you tell that DAC on direct air capture like that on plant, you tell that, sir. How do that manage our climate change? I want to
Direct
know the DAC plants you told that, sir.
DAC.
DAC.
Okay, DAC was a Department of Agriculture and Cooperation, Ministry of Environment. This Ministry of Agriculture and Farmers Welfare. DAC was that, na. So they must be. They are having plants also, na. Government is also working in that. Earlier, when I was working at the NRM division as ICR headquarters, the CRIDA have bring out the contingency plan for the more than around 670 74 or so, like that districts in terms of any of these natural calamities like drought, etc. What is to be done at that time. So the government and the research institutions are also working. Like in the previous thesis presentation, also see they have also tested the most adapted growing that across 3 environments. So the scientists are also working for the drought tolerance or the heat tolerance like that material to develop breeding more for that. But my suggestion in this lecture presentation was we already have such kind of material, which are the farmer varieties, landraces, which are being cultivated in rain-fed areas, drought-prone areas, salinity and flood-prone areas are there, but their yield is less. Okay, but they are nutritionally better. But so we have to improve them and see the basmati, whatever 1121 was being cultivated, that was also taken from the farmer variety landrace was available, that has been improved, and we have gone to that extent. But that by doing that, narrowing down the genotypic content favoring to one direction, we lose several genes which are responsible for the other characteristics for disease pest and climate. Those that we have to take care of while doing the breeding work, selecting such material that we have to take care of. So for this DAC was for the Department of Agriculture and Cooperation. Okay. Any other?
Hello, sir. Good morning.
Good morning.
Sir, I have doubt. Like as of now, we are having 12 to 13 lakhs plant species which are there, but in which only 3 to 3.5 has been discovered. I mean, by the taxonomists out of 13 to 14 or 12 to 14. So of which only 3.5. So only out of this 3.5 lakhs which have been discovered by taxonomists and biologists, we have seen like in the slides also, like in 3 digit itself, the number is in 3 digits. The agrobiodiversity is very small subset of this 3 lakhs or 3.4 lakhs.
Yeah.
So there is still so much, like almost still 7 to 8 lakhs is still there. So from that also we can get so much biodiversity, agrobiodiversity. I mean, for eating crops, food and agriculture. Out of
Two
those 7,
Two points you have raised. First is the identification by the taxonomists that what is being talked or the so on. I doubt whether that much number of gap still exists in at least not in India. See, how much number of new species are being reported by the BSI and JSI are very less nowadays. Okay, so that much gap I may not be. That maybe some people's they project it like that and say that nothing is done. So much is still available. We had not done. That's to which Personally, I do not agree to that point, particularly in respect to the India. Another point you raised that there may be several species, wild species, edible species, which have potential to use that in the food chain, food system also. So for that also, I would like to tell you that if we look at the current trend, changes taking place, youth are going for pizza and so many fast food items and not going to our traditional food systems, which is very less favored, but generally very rarely available in the market also as a ready-made food also. Okay, so going to that side of using them, there will be, when there will be no consumer, no demand, then there will be not be any recognition of the worker, scientists who will be working them, neither it will be demand also. So then who will be working on them also? I have served for 7 years in the Northeast, visited so many places. Every day they use these wild material, wild vegetables, leafy vegetable. Every day they are daily both type meal. They use it, boiled vegetable, boiled leafy vegetables. They use it, but do anybody working on conservation of the leafy vegetables? Okay, and in one of my scientists, they have documented around 400 species which are being used over there in 5 states of the northeastern region. These leafy vegetables are using, but nobody is working. And when you talk to me, ask to me how to conserve them, collect them, but where to conserve them, because they majority do not set seed, they are multiplied vegetatively like that. So where to conserve them and who will be working on them? Even if we come out with the nutritional analysis, nutritional value, nutritional profiling, that even that, but it is going to serve, if serve that if there is no use, no demand, no multiplication like that. So this is the situation of that part. The second part you told. I may be pessimistic.
So,
Yeah, any other?
so one more. Um, Last year we had COP27, and there was so much, like we have seen, like in ocean, several archipelagos, islands will go, they'll sink. I mean, water level is rising. So, is there a, like even in those places like which are about to extend, like those lands itself now? I mean to say, global area is going to decrease because of this sea level rise.
Rising sea levels due to climate change, rising temperature.
What's up? I couldn't get.
No, you mean that due to rising temperature, the sea level is increasing.
Yes, yes. My doubt is, so is there any explorations done over there? Because even from there, we can, there are so many wild races or relatives or forms which we can get from those places. And we are saying that within certain years, I mean, this is like emergency because they are telling within a decade itself or within few years itself, this might happen. So
See,
there must be explorations also. There must be emergency with agencies, right? They must go there and collect the biodiversity, which like our crops, whichever are there, their wild forms, relatives, or any new agro biodiversity.
yeah, see, uh, the- but immediate threat, I do not see any threat in India. But we have been conducting explorations, and almost every year we are going to the coastal areas also for collecting the germ plasm, particularly for salinity tolerant material. That's the trait specific material or the adapted material from that areas. We are going for that also. But coming to the crop varieties, definitely there may be scopes to find some new species which may not be, but that exploration we will only reveal that. Why we say exploration, we don't know what is there, and we go for that like that also. So definitely there is some possibility of getting some new material in such coastal areas, but you have pointed. But in terms of the CWR, we have already documented. Then we have already from 283, 700, 780 like that, we have prioritized, narrowed down to the 100. And that is also we are facing the difficulty in that to collect, because collection of the crop varieties mostly from the protected areas. We require permission from the CCF wildlife under the Wildlife Conservation Act 1972, which we generally don't get. in majority of the states. Okay, so that is another challenge we are facing here. That is being debated in the at every platform, in the NBPGR and other higher levels also. So that was my point, but but that kind of threat, which you say rising temperature in the sea level, is not immediate in our country. I do not see to that level. But certainly there are chances of getting some material if we go for exploring the along this coastal belt. All of that must be more than 3,000 to 2,000, 3,000 like square kilometer square, say kilometer area along the coastal belt of the India. That, but we have explored this Sundarban area, then other Western Ghats area in Gujarat, several states we have explored coastal belt area, in Odisha also. Okay.
Yes, so thank you.
Any other?